Microbiology 351 Honors at North Carolina State University presents Bacterial Motility in Plain English. Motility is the ability to move spontaneously and actively while consuming energy in the process. Bacterial motility is classified into 4 subgroups: swimming via flagella, swimming via the inflation or deflation of gas vesicles, gliding along a surface, and twitching along a surface. Bacteria move for a variety of reasons. They move in response to their environment to find ideal conditions of temperature, pH, and light.
Yay!
For example, phototaxis bacteria will respond to photons and move in that direction based on photon intensity.
Yay!
Bacteria sense nutrients or stimuli and move towards them. This is called positive tactic response. In negative tactic response, bacteria move away from harmful substances, toxins, or waste products. Bacteria are able to change their direction of movement through a process called tumbling. Tumbling reorients the bacteria. As the bacteria approaches bad things, it tumbles more. As it approaches good things, it tumbles less. Many bacteria swim through their aquatic environments using flagella. Flagella are classified into 4 groups. A single flagella is monotrichous. Flagella located on either end of the cell are amphitrichous. A cluster of flagella located at one spot on the cell are lophotrichous. And multiple flagella located randomly over the surface of the cell are peritrichous. Flagella are protein filaments anchored into the bacteria cells by a hook. Bacteria spin their flagella and use it to propel themselves through liquids. Counterclockwise is generally the direction they spin to go forward. When the bacteria tumbles, the flagella spins clockwise. The proton motive force is generated by the flow of protons across the bacteria cell membrane. The motor complex alone can reach a power of 6,000 to 17,000 RPM. However, with the filament attached, it can only create 200 to 1,000 RPM. Bacteria with flagella are capable of swimming 100 cell lengths per second. Another technique for moving through water is the inflation or deflation of gas vesicles within the bacteria's body. Gas vesicles are hollow structures within the cells composed of proteins. These vesicles are highly hydrophobic, but are very permeable for the movement of gases. Many bacteria require sunlight or oxygen to undergo important cellular processes. By inflating or deflating the vesicles within the cells, the bacteria can control how close they are to the surface of the water and how much sunlight or oxygen they are exposed to. The narrower the port in a vesicle is, the better it functions to propel the cell up or down in a liquid environment due to its critical pressure. Use of gas vesicles in bacteria allows them to ensure that they are in a location for optimal growth and survival. Gliding is a form of movement used primarily by bacteria which live in areas with low concentrations of water, such as soil, biofilms, and microbial mats. Gliding is defined as the movement of a cell on a surface in the direction of the long axis of the cell. Gliding may be characterized by the independent movement of a single cell or mass movement of a swarm of cells. Gliding is believed to use a wide variety of different mechanisms and is grouped by the way the movement appears, not how it happens. One example of a gliding mechanism is that used by myxobacteria. Myxobacteria use a mechanism of gliding that involves a series of adhesive sites that move along tracks in the outer membrane of the cell and can adhere to surfaces. There are many proposed mechanisms for gliding. This is only one example. The last classification of motility mechanisms is twitching. Twitching is described as an intermittent or jerky movement. Think of a caterpillar. Like gliding, twitching motility may be found in independent cells or in swarms of cells. Also like gliding, twitching motility in different types of bacteria is likely controlled by a variety of mechanisms that are classified as twitching based only on the jerky movements that are observed in the bacteria. One type of twitching motility involves the extrusion and retraction of type 4 pili. Bacteria send out their pili, which hooks the surface on which they are moving. They then retract their pili in, propelling themselves forward. This is only one of many proposed mechanisms which account for twitching motility. Using these motility mechanisms, bacteria are able to get to where they need to be to perform the myriad of important things they do that make our world work. I'm Kristen Berger.
And I'm Wesley Yang.
And this has been Bacterial Motility in Plain English.